ends meet and reduce fertilizer inputs. Another thing that society has been focusing on, and in this case, I would say this is more of an opportunity for a grower than it is a worry, is climate change and how agriculture might be able to mitigate climate change. So one trend we're seeing is that in our land-grant universities, especially in the Midwest, we're seeing a lot of grants, federal grants for research into ways to mitigate climate change. The other thing we're seeing is, really big companies making carbon neutral pledges. And in some cases, they can do that by processing farm products through their systems that are produced more sustainably. The other thing that we think there's an opportunity to do for companies that, say, Microsoft, for example, doesn't actually produce a ton of physical product, right? But they might need to actually just mitigate their own carbon emissions from the way they work. They might want to use carbon credits that are purchased, right? And so we have these markets for carbon credits, and maybe farmers could be the guys selling those credits to the big companies. So we think there's an opportunity here where carbon credit markets could become an economic opportunity for growers in the Midwest. And I just want to, mention, I've talked a lot about carbon because that's what we all talk about, right? That's the big one. But we think there's also potential for nitrogen and phosphorus to have a carbon credit market style situation too. And so overall, there's a lot of opportunity in this space of carbon credits, we think, for Midwest agriculture. In addition, there are some new and emerging markets for products that could be produced on the Midwest farm. So I'm sure you guys have heard of the huge interest in plant-based proteins and the stock market's reaction to plant-based protein products. And in fact, you can see some of the big packing plants and processors like Tyson's and Smithfield getting into the plant-based protein market. So this is something that we think could be a great opportunity for growers in the Midwest. In addition to that, just the general global demand for protein, whether it comes from a plant or it comes from a plant that is fed to an animal, that's on the rise. So protein crops are going to be something that we think can help our growers get back to profitability in the future. So I kind of laid out for you guys the scenario in the Midwest, right? Our farmers are struggling. We have some sort of ecological issues happening. We have some market opportunities we can tap into. And so when we sat down at Corteva and said, how are we going to help our growers meet this challenge? We came up with a mission statement. And that statement is written here. We can restore profitability and sustainability to the farm with cropping systems that incorporate new market opportunities, climate mitigation efforts, and the concept of circular economies. So I just want to call out two parts of this mission statement that I've spent a lot of time trying to understand in our team. And one is, what is a cropping system exactly? And so a cropping system is the crops, the crop sequences, and the management techniques used on a particular farm over a period of years. And so we have a cropping system right now in the Midwest, and that system is we plant some corn and we fertilize it, and then we plant some beans, and then the next year we plant some corn, and then the next year we plant some beans, right? So that's our system. And we do breed for that system. So we know that we can get insect and disease pests that build up in those systems. And so we've, used, GMOs, right, to solve part of that. We use just normal breeding for, say, disease resistance to solve part of that. But as a cropping system becomes more complicated, or if you want to dig deeper into it, those are kind of superficial targets. And we're thinking about what might be the other targets for breeding in a cropping system. The other concept that I just want to touch on and make sure everyone understands before we go forward, because I didn't know what it was at all when we started, is this idea of a circular economy. So the idea of a circular economy is that you treat your system, whatever your system is, like a ecosystem, and you design out the waste in your ecosystem, right? So the goal is to move from less of a produce, consume, and discard model to a model where we optimize the use of the resources in the system, and then we reuse those resources wherever possible in that system. So these are kind of the two principles we moved forward with when we decided we were going to work in cropping systems. And so when you think about the Midwest cropping system, and you try to think about what waste we can manage in that system, so optimize the resources that we have and make sure that those resources stay in the system over time. The major sources of waste and opportunity that we could identify were light. So, I mean, the amount of photosynthesis that can happen in the Midwest during a growing season is enormous, right? So we have a lot of opportunity to use light that we're not using throughout the year. The other place where we could manage better is water, whether that means managing too much water or managing too little water, or optimizing the crop or the variety that we're planting to fit the water that's available in the system. There's a lot of opportunity to work on in water. Soil is also another place where we have waste, whether it's soil erosion or just not using the soil that's there for the whole of the season. There's an opportunity there. And then we also waste synthetic nitrogen. So our growers will put on a nitrogen every year, and some percent of that will be lost out of the system, which is bad. We just talked about how that's bad for water quality. It's also bad for farm economics. So in the end, after we considered all these sources of waste, the solution that we think might be the best solution to making a circular economy style cropping system in the Midwest is to introduce more double cropping opportunities. So what that will do is help you use more of the season, the tails of the season that we're not using with our current corn and soybean crop. And it should also, if you can get a crop to overwinter, it should give you some opportunities for holding soil and getting value from ecosystem services. So we know what we want to do, and then we stepped back and said, okay, what crop might be the best fit for that? And this is where we came up with peas. So peas have a short growing season. They have overwintering potential. They have very high protein compared to many other plant grains, and lots of markets as a result of that, both for human protein and for use in livestock rations. Peas fix nitrogen because they're a legume. And then as a breeder, one of the things that I advocated for from the start was to use peas because they're a diploid, which is always easier to breed with, and they have a pretty robust genetic resource. So peas seemed like a pretty good space to start to explore. So what we did in 2019 was we got some peas and we planted them in one location, it just kind of in a bulk strip. And we called up some folks out west in the traditional pea growing areas up here in Montana, North Dakota, Idaho, Washington State. And we said, how do we grow these things? And so after talking to them, we determined we should start planting in kind of the end of March, beginning of April. So a little earlier than we would normally plant corn. And then we would expect to harvest those peas in the end of June and beginning of July, well before we would ever harvest corn or soybeans. They told us to plant to moisture, so we did that. And then they told us that to make sure we inoculated these peas. So we do not inoculate soybeans in the Midwest anymore. And the reason is because the bug is native now to our soils. Peas are new and they need a different bug, so we have to make sure to inoculate. So we did that. The other thing they told us was to make sure that we drilled these peas on 7.5-inch rows. So these are much narrower rows than what we are used to planting our crops in the Midwest. And we also were told to plant at a much higher population, kind of raise some eyebrows when we saw 320,000 plants per acre. But the reason that you need to do this when you're raising peas is to help them grow the best and be able to be harvestable. So in this picture in the corner, you can see that this pea has a lot of little tendrils kind of coming off. So there's some leaves and obviously a pod, but there's all these little tendrils on them. And what those tendrils do is wrap around the plants next to each other. And then those peas are able to grow taller, and then you're able to harvest them standing. So you don't have to swab them and then come back and harvest. So it's an efficiency of harvest issue, as well as a kind of a yield and plant health issue. The rest of pea production looks a lot like soybean production. So that's a positive. Our growers, they know how to raise beans better than anybody. And so we were pretty positive on this pea idea. So we planted them, and we actually didn't harvest them because when we asked for a combine in July, everyone was like, what? We don't use combines in July, and there weren't any ready for us. So instead of actually harvesting, we took some subsamples and we estimated what the yield would be. It seemed respectable. The other thing we did was we invited Kevin McPhee from Montana State out. He's a pea expert. And we had him stand in our peas and tell us, what do you think of these? And he kind of gave them the thumbs up and said, those look pretty good. We didn't get smoked by a bunch of diseases, so it seemed pretty positive. And we then decided to move forward with our plans to work on peas. So the next step in the process is we think we can grow peas, but they're supposed to fit into a cropping system. So what might that cropping system look like in the Midwest? And so we came up with several options. So the first one is a service crop. And all a service crop is a fancy way of saying a cover crop. And we wanted to say service crop because in Iowa and most of the Midwest corn growing states, when you say cover crop, you mean winter rye. And the rye gets planted in October, very late in the season, and it gets 2 inches tall. And then it stops growing for the whole winter and doesn't grow again until the spring. It's mostly there to hold the soil and be a nitrogen scavenger. So it does some good, but the things that it does are fairly limited. And so when you start talking about a cover crop option that gets planted in July or August, it really opens up lots more species options. And so you could plant that crop to suit whatever the problem or problems were on your farm. So you could plant a cover crop that would address compaction, that would address, maybe a nematode overpopulation. You could plant a cover that does nitrogen capture, one that builds organic matter, or a set that fills any of those needs. The second idea that we had for a post-pea crop is a biomass crop. Right now, this mostly means something to feed to cows, to graze cows or sheep or some kind of livestock on. And that summer forage is an easy one. That summer forage could also be an overwintering forage, right? You could get a nice alfalfa or a perennial clover crop planted in that time frame. The future on that biomass crop might be for biofuel stocks, right? So if we start moving to more biofuel type options as a nation, there might be a lot of option for growers to provide those stocks. The final thing that we would like to do, and this is kind of a little bit of a wilder idea, it's going to require a little more work to get there, especially in the upper Midwest, is we'd like to put a double crop out. Right now, I think we could do this with winter wheat probably, or maybe with winter canola if it was hardy enough. The other things we would like to try is maybe a really early maturity silage corn or soybean. So we have a lot of opportunities out there for cropping system components. So we have these ideas, and we sat down and thought about all of the ways that these cropping systems might change the farm ecosystem in the Midwest right now. And so if you compare the way that a pea, either service crop or a pea forage crop would compare to a corn or bean situation, what we determined is that the peas would fill a different economic niche, this pea system. So it would get the growers into new markets for plant protein and potentially for ecosystem services. They would fill a different pest niche. So there are some overlap in pathogens between soybeans and corn, but between soybeans and peas, but because the peas get planted earlier and harvested earlier, we think they might be able to escape some of those pathogens like white mold, for example. Peas and soybeans have similar types of pests, but they have in some cases, they're actually different species. So they have different nematode pests, for example. And just generally planting some kind of a pea forage rotation would interrupt some of our bigger pest cycles. Corn rootworm or cyst nematode could be interrupted with this new cropping rotation. The peas and that forage would also provide a different livestock feed situation. They would expose the grower to different weather risks during the key times of flowering and grain fill. That would happen earlier than it happens for corn and beans. And then the thing that gets most of our growers really, really excited, but we never thought of this until we brought this in front of farmers, is the opportunity to move their labor across the season. So right now they have huge labor needs at planting and at harvest for corn and soybeans, which are happening at the same time. And if they introduced peas, they could spread that labor through the season a little bit better. So that was something that our growers were able to contribute to the farm ecosystem concept that we didn't think of. So we had a plan and we executed on it. This summer we grew about 13 locations with 10 commercial pea varieties as a trial. So these were just strip trials in Iowa, Missouri, and Illinois. And the answer to the question of can you grow peas in the Midwest, not just in one strip plot in one location in 2019, is yes. So our average yields were 39 bushels to the acre. And at first I was pretty bummed out because I wanted these peas to yield like soybeans, and 39 is not equal to 60, which is what we like to see on a soybean. So I was kind of bummed, but when we compare how we did in the Midwest with how the peas grow in the traditional pea growing regions, we're actually pretty competitive. You know, in Montana, North Dakota, and Canada, we're right there in the ballpark. So that was positive. And the other thing that we felt pretty positive about was that for every variety and at every location, there was a particular combination of field location and variety that yielded a lot more than the location average. And so there's an opportunity, we think, for variety selection and site selection to optimize the commercial peas that are out there to make them a more high yielding option for our growers. The other thing we learned was that when you do something for the first time, this is not shocking, but when you do something for the first time, you don't always do it right. And so we spent all of this last summer, observing how we did things wrong. And one of the things that we did poorly was we went into these environments in a no-till situation. So we were planting peas on ground that had corn growing on it in 2019, and corn leaves a substantial residue in the field, especially in a no-till situation. And so we came in with these peas and we planted them, and often we were planting the peas right into the old corn tissue. And so the pea was not getting good seed to soil contact. And so our germination was down. And what we found was reduced plants per acre, so a reduced plant population. And there's a pretty high correlation between plant population and yield. And so, so we have some ideas of how to manage this. One is, adjusting our equipment to do a little bit better residue management in these no-till environments. And then we're also going to try to put some seed treatments on in case, that cold and wet condition is a little different than what these peas normally see out west. So we felt pretty positive this year with the yield data that we got. And so our next step, and what we've been doing of late, is just thinking about how we would actually breed peas. So the good news about peas is that there's a huge genetic resource for peas. They're diploid, like I said, so that's good news. Always easier to breed with a diploid crop. They do have a huge genome, but that's something we think we can work around. There's a public map, a SNP chip, a reference genome, a model species, all the infrastructure that you need to do, genomics and molecular breeding. There's also been work done discovering major genes for diseases in flowering. So there's a very strong public research effort in the pea sort of discovery breeding space. In addition to that, there are lots of good accessions in the GRIN collection, and they've been characterized and categorized into dry spring, dry winter, fresh, and forage peas. And there is plenty of diversity left in those varieties. And so we think there's lots of opportunity for breeding. And like I said, we have this new US pea breeding consortium, which is pretty much public pea breeders. Many of them are sort of new to peas, so that's exciting. And then me, right? So we're the pea breeding consortium, and we have a lot of ideas of research projects that we can do together, and that is really, really exciting for us. So after 1 year, and this knowledge that we think we can do breeding, we decided to figure out what would our breeding targets be. And so diseases, the first disease on the scene in the Midwest is powdery mildew. And by God, it was intense. So you guys can see in this picture, you're either resistant or you're susceptible. And if you're susceptible, it's a bad deal. So the story on powdery mildew is that if it comes in late, it shouldn't affect yield. And what it does is sort of makes the plant rubbery so that the seeds can't dry uniformly throughout the field. So it's really more of a harvest issue than it is a yield issue. In our locations, we saw a pretty sizable difference between our susceptible varieties and our resistant varieties. So we're wondering if our powdery mildew didn't come in early enough to actually affect yield. So that will be one of our first targets for breeding, will be to ensure that we have powdery mildew resistance in our varieties. I talked about stand establishment. That's obviously a target. If we cannot fix it with agronomics, we will try to breed for stand establishment. I'm really hoping we can figure that one out with agronomic practices, because we've tried to breed for stand establishment in corn, and it's really challenging. So so I'd like the story to be that we are addressing this with agronomy instead of with breeding, but it's a potential breeding target for us. The other target that we thought we would have would be flowering, and so there's some good reasons for this. So peas are a long day photoperiod sensitive crop, and that means they should be planted in the spring. But if you could shift that to make them look like soybeans, maybe you could plant peas in the second half of the growing season instead of the first. The other thing that's could be a really nice target is indeterminacy is always a benefit for stress tolerance during flowering, right? So if you flower over a long period, if at one point in that flowering you have some stress, that's okay. You can recover from it on the other sides of that stress. And then, something very simple is, could you have earlier flowering peas be earlier maturity, right? Could you use those two traits together? So when we went out and selected our entries for our experiments, we made sure to buy peas that were described as early, mid, and late flowering. But what we found this summer is that early flowering peas flowered on Monday, late flowering peas flowered on Friday. So there was just not much variation either among the varieties at a location or even across all of the locations. We planted over a period of 3 weeks in 3 different states, and there were only 10 days of difference between the first and last flowering variety at all of those locations. So if we want to breed for flowering time, we're going to have to do that in a different way, either with some kind of really targeted, editing, mutation breeding, something like that, or maybe with some accessions. And we did grow some of the Grin collection in our nursery this summer, and it looked to me like there was more variation in flowering time in that collection than there is in our commercial germplasm. So another breeding target we thought for sure we would have was heat tolerance and yield stability. So the conventional wisdom is that as you move peas from the northwest to south and east, you're bringing them into more heat and breeding them into more humidity. And so the heat is bad for seed production, and the humidity is bad for disease development. And so we were pretty worried about that. We thought we were going to have some strategies for addressing those issues. But I don't feel like our data this year is very compelling this way. So in this plot here, you have all these little colorful lines, and each colorful line is one of the different varieties. And then on the x-axis is each location. And the locations are moving from the south. So our Missouri locations are on the left, and our northern Iowa location is on the right. And then on the y-axis, you have yield, right, from lowest to highest. And so I think when you look at this plot, you could tell a story that as you moved from south to north in your locations, your yields maybe went up. So then we broke it down and said, okay, why was that happening? And so we looked at how many reproductive features, so flowers or pods, we could see at each of those locations in north to south. And I think the weak relationship got weaker when we looked at that data. And then when we went all the way to looking at how many seeds were aborted from the pods, likely due to heat or drought at these locations, you lose that relationship entirely. And so what we think is that the peas were not stressed by heat. And if anything, it was something about day length that affected them as they moved south. They weren't able to put on as many flowers because they had shorter days sooner. And then when we did the same exercise for longitude, moving west to east, we saw no relationship at all. And I think, you know, the important thing to remember about this data is that one, it's 1 year, which is always risky. Two, I talked to you guys about, you know, we had some stand issues in these plots. So I would say that, you know, 1 year of, you know, sort of shady data, it does not mean we should draw a conclusion. But it's a good sign. We felt good about it, and it's guided us in making plans for next year. So next year, we're going to clean up our plots, make sure we have good stands in them. And we're also going to add some locations. So we're going to go into new irrigated environments in Kansas. We're going to go down to the Delta, where we say it's hot in Iowa, but like you guys in Florida know, it can be a heck of a lot hotter. So we're going to sample those environments. And then we're going to move into drought and sort of high plains acres out in Nebraska with the collaboration with the University of Nebraska. So the final breeding target that we know we were going to have is protein content. So heads up on this one, is, we don't have data yet. We haven't gotten through processing our samples yet, but we know that this will be a target. So peas have high protein content compared to lots of other plant proteins, but not the highest. The thing is, they bring a lot of other good benefits with them. They're vitamin rich, they're a complete plant protein, and then especially important is that they're hypoallergenic and they have no phytoestrogen. So this is part of the reason they're really growing in the plant protein product markets. And as far as livestock feeding, they have an advantage over soybeans, especially because they don't have trypsin inhibitors, so they don't have to be heated and processed before they're fed. So these are great opportunities, both for our growers, for the processors, and for the feeders. And so what we'd like to do is get the protein content up as high as we can get. So what the published data says that most commercial varieties are sold with a range of protein from 24 to 28%. And so many of them are on the lower end of that. So we can, you know, move up there. And then a small study done to sample the GRIN collection for protein suggests that we can move it up even a little bit higher there. So this is a trait that's easy to phenotype with NIR, and so that's, you know, pretty positive. And so we developed a breeding strategy. One that is kind of leveraging what we know about soybean, which is that there's a negative correlation between yield and protein in soybean. That hasn't been observed quite so robustly in pea, but biologically it just makes sense. So we're expecting that to happen. But we know that there have been QTL identified for protein content. And we know that the bean breeders have been able to solve for this problem. So our pea consortium is going to work on a project to look at G by E for protein content. And we're hoping to, you know, have some really good success in that space. I spent a lot of time just now talking about protein, and that's going to be the first problem we attack for pea quality, but we're also going to work on improved amino acid content and flavor for these plant protein processors in the future. So we've been talking through this whole time about spring peas, and I wanted to talk briefly about winter peas. So winter peas are planted in the end of September. So, you know, after an early corn crop or bean crop might come off, and they get harvested around the same time as a spring pea. And winter peas can do everything that a spring pea can do, everything that we just talked about, but they do it while acting as a cover crop over the winter. So that's good for the soil, it's good for ecosystem services, it's just generally good for your cropping system. So this is where we really want to go. And if this is where we want to go, we have to add freezing tolerance to our breeding targets. So it seems like could be successful. We have variation in the germplasm. We have QTL and candidate genes, at least for frost tolerance, and some indicators for freezing tolerance. So what we did, and this is super exciting, what we did was start a collaboration with Kevin McPhee at Montana State. So Kevin has been working a winter pea breeding program up in Bozeman for a couple years. And so you can see in this bottom left picture, his winter pea experimental. So there's some differences in survival in the spring. And then next to it, you can see a comparison between the winter experimental and then a spring pea variety. So there's a real decided increase in winter hardiness in Kevin's winter pea populations. And then this next picture over is those populations grown in our Dallas Center, Iowa nursery. It was a beautiful day in December, and I ran out and I took some pictures of those peas growing. So we're feeling pretty positive on the potential for this collaboration and for winter peas as part of our cropping system. And then this final picture I wanted to show is a picture of a freezing chamber. And so obviously the best way to phenotype for winter hardiness is in the field, right? You cannot simulate the conditions of the field in a chamber, but you can do some pre-screening, and you can do it a lot faster than you can, obviously, by waiting for winter to come. And so we just started our first winter chamber trials a couple months ago, and they're looking pretty positive. Okay, so I just gave you guys a whole brain dump about grain peas, and now I'm going to change focus a little bit and talk about using peas instead of for grain but as a cover crop, as a nitrogen source. So our reasoning here was to use peas to help our growers move to a reduced, fully in-season synthetic nitrogen application. So when you grow corn in Iowa as a farmer, what you normally do is you apply your nitrogen in the fall before the crop is grown. So in November, you see a bunch of guys driving around with a big tank of anhydrous gas behind them. And so that nitrogen sits in the soil and then it's going to be available for the corn crop the following year. So this has some negatives to it. So the first part of it is just the fact of synthetic nitrogen is not great for the climate. So we use a lot of fossil fuel energy to make that nitrogen. That nitrogen costs the grower money, and 20% of it on average is lost in any given year. So that's bad for the grower, his bottom dollar, but it's also bad for the nitrogen that's leaching out into the groundwater. And then the other thing about synthetic nitrogen that is not as good as sort of a natural nitrogen that could be produced by a plant is that you don't get the carbon sequestration benefits or the soil erosion mitigation benefits that you get. So our idea was to replace some of the synthetic nitrogen in the system with peas and to move to a fully in season system. And the idea is based on a research that was done out east on fava beans in a sweet corn system, and that looked really positive. So we did a small trial at a site we have in Villago, Chile, where we planted peas before corn crop. We planted a mix of peas and rice before corn crop, and then we did traditional fertilization. And you can see in this picture that the normal fertilized corn looks the greenest and the biggest and the fastest growing and the best, right? But the pea cover crop isn't far behind. So that gave us some hope that the system could work. So we started thinking about, what's the research we need to do? And a lot of agronomy research that needs to be done on planting method, density, are we interseeding these? When are we planting them? Are they going to be overwintering? If they are overwintering, do we need to suppress them in the spring? When they break down and contribute their biomass to the soil, when does that nitrogen become available to the crop? And how durable is that system in various different soils and weathers? So assuming we can figure all of that out, the system we're expecting to develop is one where growers plant their corn with a starter fertilizer. Then they rely on this pea nitrogen from the pea that was grown the previous season. And then they do a side dress application in the end of June, and that will carry the crop through to the fall. So this is the idea for the system. And then we started thinking about what would be the breeding targets in this system. So what we think is that the biomass is going to be the key to producing the most nitrogen in these peas. And so if you can see on the left, you have a picture of a forage pea with lots and lots of leaves and not so many tendrils. And then on the right, a picture of more of a grain pea where there are a lot of tendrils and not so many leaves. So we're thinking we need to target a forage type pea. There isn't much of a footprint for retail forage pea sales and almost none for breeding forage peas. But there is a national legume cover crop breeding effort that's being led by Cornell and is being carried out across the country in this cool farmer breeder system. And we're lucky because it does include some northern states that are going to have a focus on winter hardiness. The other thing is that there's lots of variation in the GRIN collection for leaf type and plant type and biomass. So there's potential in the germplasm to get something done here. And our breeding targets will be high biomass, both above and below ground. Right now we're focusing on above ground because I've dug some roots and I don't want to dig anymore. That's awful. So we're going to try for above ground biomass breeding. We think we might have to think about shade tolerance or intolerance depending on what planting strategy we end up with. Frost tolerance and vigor at the tail ends of the season is probably going to be important. We also talked about some things that are kind of wilder and more futuristic, but they're really exciting. So could we breed for nodulation rate and stability of nodulation rate? Or could we even breed for varieties that are customized in their C to N ratio to fit the best in a particular soil type and weather? So those are cool ideas. We're definitely not working on those yet, but that's something we're thinking might be an opportunity in the future. Okay, so I'm just going to wrap up now, and we'll have some time for questions. I hope that I've been able to show you guys how we're rethinking breeding in the context of a cropping system as a circular economy. And so when we do this, it's exciting, it's new, because we have to consider how to optimize the whole farm ecosystem. And that requires some of the traditional breeding tools that we're all familiar with, like genomics and good phenotyping, physiology, things like that. But it also requires scientists thinking about, ecology, micro, plant, and animal biological interactions, meteorology, systems engineering, marketing, farm profitability. So we need the economists in with us. And then sort of downstream environmental effects. And we have to think about all of those things over time. So it's a really new and rewarding challenge. And one of the things we've learned as we've gone through this is how important the public-private partnerships are going to be in getting this done. So those have always been important, but this seems to me to really bring it to light, that we really need to work together with our partners in the USDA and with the universities. So I guess thanks guys for listening. We can have hopefully a little bit of time for questions here now.
Thank you, Dr. Lira. Good afternoon, everyone, for whoever joined this afternoon session. I'm Lushan Gimire, a PhD student under Dr. Patricia Munoz's Blueberry Breeding Program. And today I'll be moderating the Q&A session for the rest of the day. And moving towards questions, we have a couple of those. Let me proceed with the first one. So the question here is, are there any current efforts to use new breeding tools to combat climate change, and the tools like GMOs or CRISPR, and what are the opportunities and challenges around that?
Yeah, so that's a pretty big question. So what I'll do is bite off the one thing that I've been thinking about lately, right? So I think there's a lot of opportunity for CRISPR in some of these crops, right? Especially this protein issue, right? I think CRISPR could go in and clean that up like right quick, right? And the problem is the acceptance of CRISPR as a GMO or not GMO, right? And it becomes increasingly important when you're thinking about a crop that could actually go directly into a person. I think when we see people react to GMOs in corn, for example, they're worrying about something that's going to get filtered through an animal before it ever gets to them. I think that we really need to resolve that question before we would think of doing CRISPR in grain peas, just because they have a human as a kind of immediate next user. But I do think one of the things that we've talked about that I think could be really exciting is the potential for using CRISPR in a crop that never gets harvested. So if you could use CRISPR to modify something important in a cover crop pea, I wonder if you would have less regulation in that. We don't know, we haven't started exploring it, but it's been something that we've tossed around as a potential really positive about breeding for a cover crop, is that you might be able to get around some of those GMO fears.
Thank you. We have another one. So how sustainable is this crop rotation system for smallholder farmers? And do you have a way of getting feedback from them?
Yeah, so, right. So, it depends on the definition of smallholder, right? So if you're thinking of the guy with 50 acres, that's probably not going to work for him. And it's a shame, but with 50 acres, I think it's better to have someone breeding tomatoes or growing peaches, or kind of the traditional crops that you think of for small farms that you guys have been talking about today. But I do think that this has a special relevance to what we think of in the Midwest as a small farmer, which is, honestly, guys, 1,000 acres is a small farmer in our systems. And this is extremely feasible for a small farmer in our systems. And actually, the increase in the potential profitability and the diversity of income sources and the value to livestock, it really actually is targeted to those growers. It could work really well for them. So yeah, it's not going to help the 50-acre guy, right? But it will help the smaller of the Midwestern growers, I think.
Thank you. We have one more question here. So it's by Joshua. He's asking, historically, we've seen big agricultural companies buy smaller, diverse seed companies and then strategically focus on money making, which is kind of unfortunate. But if we ignore goodwill motives for circular agriculture and sustainability, is it in the public arena, or is it companies' strategy for diversification of products that more motivated that more motivated the start of this P project? And he just wanted to see, like, why hadn't this been done sooner and all that.
That's a fair question. Yeah, it's a fair question. So what I think is, right, we are a publicly traded business, right? And if we don't make money, we get hassle from our shareholders. So we have to make decisions that have a market. And I can assure you that, it's been really interesting for me that there's a lot of goodwill and good heart in the people that work in research at Corteva. And all we needed was an opportunity to let it all out. And what I think is that in the last 5 years, I think society is changing, and this move for climate mediation and for better quality foods and better environmental protection, I think it's growing and growing. And so as that happens, there starts to be an economic opportunity in working in that space. And so I think the reason that it took us this long to get here is because there was no economic opportunity prior to now. So I really do think that things are changing for the positive, and I hope we're going to be able to put our growers in a position to, I guess, take advantage of that, right?
Thank you. And we have one last question here. So how has your 10 years of corn breeding experience played into this new venture? And was there significant overlap, or did it require a lot of adaptation on your part?
Yeah. Right. So it's amazing. You guys will find I graduated a long time ago, and I hadn't thought about a varietal crop. I've only thought about hybrid crops for a really long time. So I had to so in that way, it took me some time to kind of redirect my brain. But I think a breeder is a breeder, and the things that are important in developing varieties, the principles are the same, right? So I do think it helped me. I think that being so deeply embedded in the system that we're trying to change actually gives me knowledge of what will work, what won't work, what will be an advantage, and what won't be an advantage. So it required me to think a lot differently, but it also brought a lot of knowledge of the system that I was trying to break into.
Thank you. Thank you for all the answers.
Thank you, guys.
Okay, thank you, Dr. Lira, for your presentation. I think my favorite part was when you started talking about, I guess, sort of the mistakes you made progressing through this adventure. And it's often not a side that we get to see from companies. And I think all of us graduate students here know that oftentimes mistakes are the best teachers moving forward. So it was really cool to hear that from you.
Thank you.
Okay, I'm now going to go ahead and introduce our next speaker. It's our last speaker of the day. And I had the pleasure of meeting her at last night's networking session, and it was certainly a wonderful experience. Hearing about her journey through the education and research positions until she ended up at Clemson University was truly encouraging, and it made me feel as though all of us undergrad and graduate students and postdocs we'll eventually find a way to make a name for ourselves in this wonderful little community of plant breeders here. So Dr. Ksenia Gasic works on the development of high quality, disease-resistant peach varieties adapted to environmental conditions of the Southeast US using both traditional and molecular breeding techniques. She specializes in characterization and utilization of the peach genetic diversity in land races, gene bank collections, and elite germplasm for germplasm enhancement, and utilizes marker or QTL discovery for the traits of interest. Dr. Gasic is continually looking for ways to improve her program, including the development and utilization of genome technology to improve breeding efficiency. I know we have a lot of peach fans out in the audience today, so I'm not going to make them wait any longer. I now present Dr. Ksenia Gasic.
Thank you, Julian, and thank you for inviting me to give this presentation. It's an honor to close this great symposium, and I must admit I did not have time yesterday to listen to the talks live, but I watched the recording today. Thank you for sharing that link for the Facebook, and it was really impressive, the number of speakers and the talks, very interesting stories. So now it's the time for you to see something unusual, let's say, in the world of breeding, especially in the world of peach breeding, for starting something from scratch in the peach breeding program. And so that's what I'm going to try to share with you, my story and how I started the peach or restarted the peach breeding program at Clemson University. So the whole presentation will go similarly in the ways that other speakers talked. I would share with you the background, my background, and the background of the program, the challenges that the breeding program for peaches faces, accomplishments from the moment I started the program till last year, and these accomplishments through discovery and application and what lies in future. And through these stories, I hope you will see the how it went for me and what you can expect going on in your careers. So peach breeding at Clemson University was dormant for 25 years. I was hired in 2007 and started in 2008 in January. So at the time I joined the breeding program at Clemson University, there were lots of existing resources in peach research, but there was no breeding program, active breeding program going on. So Muscle Fruit Research Farm is an excellent resource, which is just 10 minutes away from the main campus. And we have about 100 hectares or 200 acres of land. Majority of the research was done on peach, but there are some other crops just to have our students experience other crops. And we also have muscadines that Margaret likes and I don't. So I'm hoping Margaret will develop some good ones that you don't have to spit around. There was excellent peach germplasm. Clemson is a unique place that has a whole team working on one crop. So I was kind of final puzzle to the team because we have an excellent horticulturalist, virologist, the plant pathologist, physiologist, and horticulturalist, and so and geneticist. And so I'm kind of a person that combines all of their knowledges and try to apply it in developing of the new peach cultivars. What I brought to the position was knowledge and experience in conventional breeding. My whole schooling I finished in Eastern Europe, used to be Yugoslavia, now it's Serbia. And I got my molecular experience and knowledge through postdoc in University of Illinois at Urbana-Champaign. So after my postdoc endeavor, I was thinking, I don't like to be in the lab. I don't like to spend my days in the lab coat and pipetting all day and looking at the screens. And I love being outside and I love to do the breeding and walking through the orchards. And so when the position at Clemson opened, I applied and here I am 10, 13 years later, trying to tell you how how it went. So at the time when I started the position, what we had for the resources available, and still excellent resources, a genome database for rosaceae was established, and which houses all of the molecular genomic genetics phenotyping data for rosaceae species. And at the time, the markers that were used were RAPIDs, RFLPs, SSRs were just at the beginning, linkage maps and QTLs. So resources today, we have genome sequence of peach. We have also the SNP array, and that was recently improved from original 9,000 SNPs now to the 16,000 SNPs, and many more maps, QTLs, genome-wide association studies, and many more opportunities to do the research. So none of this was available when I started. And as Julian told you, my goal of the program was to combine the traditional breeding with the molecular tools to speed up the selection process. So one of the important things is to say, why is peach important for the South Carolina? Well, South Carolina is the #2 in the fresh market production in the United States. We are second only to California. Peach industry grosses about $80 million annually and is produced over 17,000 acres and even more and growing. This star reflects where the Clemson University is, and the little peach icons throughout the state show where the peach is grown, with the major peach area being this section here called Ridge, and leaning onto Georgia, which is also important in the peach production, and there is a Georgia peach production area. So we have a really similar climate. So as I said, the breeding program rejuvenated in 2008 after 25 years of dormancy, which meant that what I found at the farm or in the research available for me, or resources available to me, was excellent germplasm and excellent community. But I had to start from making my own crosses and developing the hybrids. So first thing I did, I communicated with the breeders. And I was lucky that in Byron, Georgia, Dr. Oki was just about to wrap up and retire from his breeding career. And he was very welcoming to accept me to visit him. That was one of my first things when I started the position, to visit Dr. Oki and talk about the problems and issues of breeding for Southeastern region. In addition to that, I went to University of Florida to visit Dr. Jose Chaparro, the same thing, and to talk about his research and how that affects for the climate of the Southeastern Florida, and so on. So at the time, Dr. Oki published a paper about the Eastern United States peach breeders. And in the paper, he actually listed some of the qualities that he thinks breeder needs to possess, which is the vision as an ability to see what is needed and what is feasible, passion and devotion to the job daily and over the years, and the willingness and ability to work for a distant payoff. Discipline in being able to keep in mind and execute the many steps to get there in an efficient way, and risk as the willingness to release embodiment of the vision, which must then make it on its own in the orchard and market, but might also fail. So I hope through the presentation, you will see how I measured up to these breeder qualities. So as Julian said, the goal of my program was to develop high quality disease-resistant peach varieties adapted to environmental conditions of Southeast, particularly South Carolina, but then more and broader. So main focus at the beginning was to combine the disease resistance with food quality, because if you can imagine, when you start a breeding program from scratch and there are so many high quality and successful breeding programs, both public and private in the country, it's really hard to find your place. So what was unique about the South Carolina and Southeast region was our environment that was very humid and prone to many diseases and pests. So that was one of the things that was lacking from cultivars coming from, let's say, California private breeding sector, because they never have the opportunity to test them in the environment like ours. And once they come here, you realize that they have a trouble being as productive and as beautiful as they are in the drier climate because of the humidity and insect pressure. So in collaboration with growers and the great peach team at Clemson University, we decided that brown rot and bacterial spot are the two diseases that are creating the biggest problem for the state. Since those were the initial 5 years. So when you get a position at university, you go kind of in those rotational cycles of 5 years, you know, till the promotion, tenure, and so on. So after my promotion to associate professor, and slightly before that, I realized for the duration of the breeding program, you have to be visionary. You have to predict what the future will require of the new cultivars. And in that sense, you have to kind of think ahead of the even growers and producers. So I started working on nutritional quality, productivity, ripening patterns in plum, genomic selection. Recently, I started being involved in the rootstock breeding for resistance to armillaria, genome editing, and so on. And I hope I'll show some of this through the presentation. So the main challenges, which I think are across the crops, not just for the peach, climate change. And we've seen in the last 5 to 10 years that in 2 to 3 occasions, Southeast lost complete peach crop due to the fluctuation in the climate. Another thing is the consumer demands. If we look into the consumer acceptance of the peaches in the market, it's very low and it's declining because the excellent quality and the taste that we know peach can get when it's ripened on the tree is hard to get and shipped to the stores without damaging the organoleptic look of the fruit. Of course, producers need to make sustainable crop, cost effectiveness, disease resistance, and last but not least, retailer education to understand the new traits in the newly released cultivars and the opportunities for them to market them in different ways than it was normally done. So this is like a bird view of my program, which starts with the breeding, marker-assisted selection for the traits that I could do, evaluation in the field and in the lab, and we apply all kinds of tools and methods to try to shorten that cycle that can take up to 20 years. So we like to say in a fancy way that the latest innovations in science and technology are integrated in plant breeding to provide sustained solutions to production challenges and market demands. What that means, you'll see in the next slides. So this is where the magic happens. This is the Muscle Fruit Research Center at Clemson University, surrounded by the lakes. So the climate is ideal for growing peaches. That's why we chose it. The land belongs to Clemson University and then forested land as well. So we just cleared the land as much as we needed. And you can see by the boxes on this slide who's the biggest user of the land on the farm. Before the breeding program started, you know, for horticultural research or the germplasm evaluation, you basically pick the area of the land, you plant your orchard, and then it sits there for 15, 20 years. In my case, I'm planting every year. The trees are there for 4 or 5 years. Then I clean what I don't want, but those remnants, few trees that I keep are still standing there and occupying the land. So it was adjustment, let's say, to the breeding peach team to accept that I will be kind of bossing around with them about the land and availability. So in this slide, you can see, and the color-coded legend here, what the boxes mean. I was rotating around the 3 areas of the farm to plant the seedlings. So in this block here, the first seedlings planted, and now 4 or 5, 6, 7, whatever, 2016. So 7 years later, we came back to plant in the same spot, because you'll see later on, and I'll touch it just with a short remarks, that we have a lots of replant issues with the peach production, and that's why we have to rotate when we clear the land from growing peaches. So now I had to extend the advanced selection block, which is these green, 2 green boxes. And in this area, you can see that since 2017 to 2020, all of my seedlings were planted in this block only. You might ask, how come I used so much land before and now I'm just planting in this small piece of land? That's the process of using a marker-assisted selection to remove material that you don't want because it doesn't have a resistance. And you'll see that in numbers and examples later in my talk. So the challenge, the first and biggest challenge in the tree fruit breeding program is the time. Like in many breeding programs, but in tree fruit, you have that juvenility stage that you have to wait to see the first fruit. So basically in years 2 to 5, year 1, we do the pollination. Then we collect the seeds, plant them in the greenhouse, overwinter them in the greenhouse, and plant them in the field the next year. For the material that's ripening early, the seed will not germinate in nature. So we have to put it in tissue culture and germinate it, provide the nutrients so the embryo matures, store it in the, give it the cold storage, and then germinate it in the greenhouse, and then plant in the field. After planting, we have to wait 2 years till we see the fruit. So in the year 3, from the pollination, we actually have the first fruit to evaluate. Normally in the program, we evaluate for 2 years the fruit quality and then decide which ones are going to make to the second level of selection. They stay in the second level of selection for 6 to 10 years. And then we decide which ones we're going to move to the replicated trials and expose them to more environments than just our research farm at the university, and then potentially go into releasing as a new cultivar. So this is basically how that peach breeding timeline goes in short cartoon. So where we are today is here in the 10 to 15 years. In this numbers here, you can see that so far I made 382 crosses, combinations of 2 parents. I forgot to say that peach is the diploid species, which is much, much easier than for some of the polyploids that you were hearing about throughout these 2 days. And approximately 27,000 seedlings were planted in the field. Out of those, we evaluated approximately 25,000 in the period of 12 years, or actually, I should say 10 years, because the first one to evaluate were in 2010. We advanced, we have right now about 2,000 hybrid trees in seedling trials, and we advanced 650 hybrids into the second level of selection. That number is a bit high than what you would expect in the normal breeding program that runs for years, because at the beginning, I was trying to establish the germplasm diversity as well as to get the high quality cultivars for release. So kind of playing between the pre-breeding and the breeding, because I needed that diversity into my program to be able to actually select for the traits that I wanted. And so right now I have 10 advanced selections in replicated trials at 7 locations. We have 4 locations in South Carolina, 2 with the growers, and 3 throughout the state. I have less than 10, I think, selections, actually with Margaret. So she's evaluating them in Arkansas. We have them in Alabama as well, and in North Carolina. So I'm already getting a nice feedback from my colleagues about how they perform. So hopefully in the next year or 2, we will be releasing the first cultivars from our program, and then hopefully that will follow in the years to come. So the selection in my breeding program is based, of course, on the phenotypic evaluation in field and the lab, and with application of markers to decide which parents to choose to make the crosses, to make sure that we have a better chance of selecting the combination of traits that we want. Then to actually weed out the seedlings in the greenhouse before we plant them in the field, because in 2010, before I had any markers to apply, I planted about 20,000 seeds. And I had actually 20,000 seeds, about 10,000 went directly into the planting, and about 6,000 went into the tissue culture, because those mother trees were early ripening. And then through that process, you have some losses, and we ended up with the 12 to 13,000 trees to evaluate. It was a hell. In the humid climate during the summer, what I was doing the whole summer was just walking the field and eating peaches. So somebody might say, well, why are you complaining? This is a great place. It is at the beginning of the season, but after a month or 2, you become tired. So I welcome into my program the markers, at least to eliminate at the first stage with the disease resistance. So I was fortunate that my program, startup of my program, coincided with the funding of a Rosebreed project. And Rosebreed project was funded for 10 years. So basically, I had a whole community support to develop the tools and resources to apply the marker-assisted selection in my program. So that's really, I consider myself lucky with the friends and colleagues that I met through this project and with tools that were developed. So the first Rosebreed project focused on developing the tools for rosacea species and focusing on the fruit quality. And the second one included the disease resistance and combining the fruit quality and disease resistance. So within that project, we started looking at the bacterial spot resistance or tolerance in peach fruit. And bacterial spot is really a nasty disease towards which we don't have a solution. We could apply some copper sprays and some antibacterial sprays, which is very expensive, but if the conditions are right, there's really nothing you can do, and you can see what it does to the fruit and what it does to the tree. This is my student, the first PhD student that I had in my program, and he found QTLs associated with both leaf and fruit, and fruit-associated QTLs were actually with major effect on the chromosome 1 and chromosome 6. So we were successful in developing the SSR-based DNA test for chromosome 6 bacterial spot, for which we realized that is more important to have the resistant alleles than the chromosome 1 QTL location. And we started using it in 2015, and you can tell, you can see how many hybrids we tested, and the price for that test was about $5 to $6. So I paid about $20,000 to our genomic center to do these analysis. But at least I removed a bunch of hybrids that I developed through crossing and planted about 2,000 seedlings in 2015 crossing season instead of 4,000 or 5,000. In between, we actually got much better results with developing much precise tests for both chromosome 1 and chromosome 6. And to be able to do this and get the price below $1, I have to thank the University of Florida Genomics Lab that developed an excellent DNA extraction protocol for strawberry that works beautifully for peach as well, which is a crude extract. And we could use it and cut the cost really low and actually do the testing in-house on the real-time PCR machine and the regular endpoint PCR machine. So that's the answer why I have this small block of land now for my hybrids, because I'm now planting my seedlings after culling them from the greenhouse. So this year, we basically went backwards and we evaluated everything that I kept in the program, thinking, okay, maybe I like this because of this, or I can keep this particular tree, even though maybe it doesn't have all the traits that you would want for the final and finished cultivar, but I can use it in breeding. So we tested about 4,400 hybrids from last 4 years crosses, and selected about 37 to keep and to remove the rest. And the cost, as I said, was less than $1. And only highly or moderately tolerant hybrids were kept. One thing that I neglected to mention is that when I talk to the growers and to the South Carolina industry to find out what is it exactly about the bacterial spot and brown rot that they would want me to achieve in tolerance in the new cultivar, they said they're not that much interested in the leaf tolerance as long as the leaf is not that much affected to affect the next year crop, but the fruit has to be blemish free because you cannot sell it in the market. And so that's why we focused first on the fruit DNA test for the fruit resistance. And of course, we were lucky that the major loci for the fruit, the tolerance, were just two genes on the chromosome 1 and chromosome 6. For leaf resistance, we found that it's on a four or five major loci and one on chromosome 5 that's both for fruit, important for fruit and leaf, and the others were on chromosome 4 and chromosome 6 and 1 as well. So that's part of the collaboration that Margaret and I have to try to figure out if we can have the test for DNA test for the leaf resistance, or we're going to have to go and try genomic prediction if it ends up really being polygenic and across whole genome. So after we tested all of these previous seedlings and the ones that I already selected based on the fruit quality, I was pretty happy to find out that I had some excellent candidates that were homozygous resistant for the marker and that covered the ripening season from early in May to September, that were large, attractive, with a yellow flesh, freestone melting, sweet and balanced, flesh and smooth, firm and juicy. So the peach market in United States in general wants yellow flesh, freestone melting peach. And growers want red coloration, which is really hard to harvest because this peach looks ready, but it might not be because we have a types of peaches that can hang on the tree for a longer time of period and actually get better fruit quality if you're not, so to speak, trigger happy to harvest them as soon as you can, as soon as they reach the size and the coloration. So those were some of the learning curves that I had to adopt or get used to it, that you have to kind of teach the growers that with the new material that's coming out, either from your program or from different programs, they have to learn to adjust as well and not just apply the methodologies or harvest them the way they did for the older cultivars. So the next big issue in our growing area, and I think in the whole world, and it's becoming more and more problematic, is the brown rot caused by Monilinia fungus. This is a fungus that basically attacks either skin or the flesh in peach, and also can cause many post-harvest losses. Additional problem with this fungus is that it attacks the flowers and the fruit. So basically, growers have to start spraying from the bloom time and continue till the harvest, and which is not really good on their economic side. But what's even worse, even though we have a great chemistries that can control the brown rot, because the pressure on the fungus is so high with spraying throughout the season, we already have races of the or fungus that or isolate, better to say, that are showing dual resistance to the major classes of the fungicides. So when I started my program in 2008, that was the early time when our pathologist, Dr. Guido Schnabel, made the discovery that in South Carolina, we have isolates that have a dual resistance, which meant that in 5 to 10 years, if it progresses and that isolate becomes more spread out through the state, our growers will have nothing to spray the peaches with. So unfortunately, with the brown rot, it's not as nice story as with the bacterial spot, because it's much harder to evaluate. So in this cartoon here, you saw what we did harvest fruit in the field, and we realized that we have to bag the fruits to protect them from spraying, regular spraying that goes on the farm, so we can actually evaluate the true performance for the inoculation. Then we also inoculated intact skin, and we prick the skin, provide the opening for the fungus, and inoculated the flesh. And that's all work on my at that time, master student Van Fang Fu, and now she's a PhD student that's continuing this work. So what we found that in our breeding material, we have actually a nice variability in the response. Shaded areas are showing you the skin response, and these bars, solid bars, are showing you the flesh response for the brown rot. And these two families, even though the average did not give us a significant improvement, we could see that there are some individuals that have really low susceptibility to the fungus. And so what I have to mention here, the source of resistance or tolerance to brown rot comes from the feral landrace from Brazil called Bolinha. And Bolinha is really ugly, is small, green, hairy. Nobody would want that, would want to eat that. So when you do the cross with something, the germplasm like that, you end up with all combination of bad traits, and it requires several years of back crossing to actually achieve the fruit quality, with the hope that you can keep the disease resistance or tolerance present. So I was really fortunate that Dr. Oki gave me pollen from his selection that is 6th year of back crossing from that initial cross with the Bolinha, and that I used in my program to make the crosses. So these two families are from crossing with that selection and the high quality cultivars. And this is how those circled in red look like. So the pollen donor still is not anything close to what you would want to release to the market, because it has a low blush or redness of the skin. It's a clingstone, non-melting, but the size is much improved, the taste is much better, and we have some really nice selections that right now the growers in South Carolina are considering for maybe using for organic production. So there is a silver lining in this, although I'm using heavily these that are showing on this left side of the graph in my breeding program. So when Van Fang did the marker assisted, looking for the markers and the regions in the genomes associated with the brown rot tolerance, unfortunately, the story was not as clean as with the bacterial spot. We ended up with initially 32 SNPs across the whole genome, and then with applications of some multi-loci genome-wide association methods and models that are recently released and published, we narrowed that down to the 14 SNPs significantly associated with brown rot tolerance in skin and flesh. You can see on this cartoon how they're spread out through the genome. The only chromosome that did not have a significantly associated SNP was chromosome 3. And color-coded red means that tolerance is associated with flesh, and green with skin. Why this is important is because if growers can control the insect pressure, because insects provide entry points for the fungus to penetrate the skin and infect the flesh, then maybe we can have some way of managing brown rot without completely going away from sprays, but maybe reducing them to minimum that would provide growers with a high quality fruit, but also would reduce the amount of the pesticides applied in the environment. So with this result, as I said, starting with 32 and now 14 SNPs, we decided to test the genome-wide prediction, and I'm using it for last 2 seasons in picking the best selections in my program to use for further back crossing with the high quality cultivars to improve the fruit quality. So, and Van Fang is just about to publish the paper about that study. As I said in the beginning on the slides, which are the additional traits that I looked at, one of those was nutritional composition and accumulation in peat germplasm. We also did the same story, looked at the germplasm, looked at the segregating progeny. We did a biparental mapping. We also tried the genome-wide association study, and we found some really interesting regions in the in the peat genome that show accumulation and control of accumulation and composition of many of nutritional traits. Then we expanded that to look into the carotenoid composition in prunus fruit, and not only peach, but also apricot and plum. And with the idea that if we can control the composition and accumulation, we can maybe control the color, the flavor, and potentially influence the ability of peat tree to fight off the pathogens, because we know that carotenoids are the compounds that are involved in the defense system of the plants. This is my master's student that worked on peach in this project, Mark Jasinski. He finished last year. So with all this story and as I was going through, you could see that there were so many traits that we discovered. And when you look into the resources in the genome database for rosace and the international community that's publishing and releasing new information, we found out that there are regions in each genome that harbor many important horticultural traits. And you can see that for this chromosome 4 on how many different traits from fruit quality, productivity, to the disease resistance are all harbored on this chromosome 4. So the idea was through the Rosebreed project to understand how these chromosome 4 regions are being inherited and the variations within each region that directing the inheritance on the particular traits. So we could do kind of a puzzle picking. And so if you imagine that the chromosome is divided into the 14 different segments of haploblocks that are not recombining, and then you know for each of these blocks, their performance, which are featured here in this cartoon, that could go from the high resistance to low resistance or high quality to low quality. And if you have an ability to just pick and choose the version of each of these blocks or the allele that is giving you that trait, that would be the ideal approach to get the cultivar you dream of. So where this story goes, the peach belongs to the Prunus genus, and it's a diploid species, as I mentioned before. Almost every single crop in the Prunus genus, which is almond, sweet sour cherry, apricot, European and Japanese plum, has the genome sequence. So what we are able to do now that we couldn't do 10 years ago is to do a comparison between these genomic sequences, and with the QTL and marker data released for each of the crops. In this case, I'm sharing with you the peach versus sour sweet cherry hotspot on LG4, where we found that in the same region in both genomes, we have the same fruit quality traits mapped. So when we do compare those regions, we can see how they differ. And for majority of the traits that were not mapped in the cherry, but are mapped in the peach, we can actually predict if the cherry has those genes, and vice versa. For some traits that were mapped in cherry, but not in peach, we can use it to kind of surpass all of this discovery, mapping and everything, and go directly to the genomic region and try to understand how it affects the trait inheritance and exposure in the plant. So somewhere in the line of this story, we started to including looking into the prunus crops as a relative and exploring synteny and collinearity within rosaceous fruits and prunus crops, and use this nice mutant series of plums to understand the fruit softening, to try to apply that to the peach. And as you know, in the some other crops, you can have actually the mutant series like Arabidopsis, and you can test your theories. We are not that lucky in the fruit world, but for some reason, we were extremely lucky to get sports in the Santa Rosa cultivar that actually differ in the ripening time, but also in the fruit rate of fruit softening that we could apply potentially and discover what's going on and try to apply that into the peach breeding program. I'm just gonna flag over these slides. So of course, with the genomic editing and all new tools developed, we got excited, and a master student, Jonathan Windham, is trying to do some type of a gene editing to make peach plants and roses flower constantly. So we'll see how that goes. You can imagine how many data you can generate in the breeding program, not just phenotyping data, but molecular data, genome sequencing, the genotyping data, transcriptomic, and all of these previous speakers mentioned, all big data. And then you just scratch your head as a breeder, what do I do with this? So as a breeder, you are basically a person that kind of is a hub for all of these great expertises. And so the best way to do that is to collaborate with the people that are smart in their expertise, in their fields. And in my case, we have a great translational genomist, is Dr. Saski, that helps me a lot understanding and manipulating these big data. Also with the people from the genomic database for Rosace, I'm collaborating on development of the tools for collecting the data from the field and archiving and analyzing that data in a database format. You can recognize these pictures because this field book app, which is freely available for everybody to use, was developed in the Jesse Poland's group for wheat breeding. And now we are fully adopting it to the specialty crops breeding and tweaking it to fit our needs, because it's quite different from the field crops, but it's fairly easy to actually adopt it. And in addition to that, we can store that data into the breeding information management system, which is the BIMS acronym, in the genome database for Rosace, which is also free, and has genome databases for several other crops, for pulses, for citrus, for vaccinium, I think, and I might miss some of those, that each of them have this capability to actually directly collect the data electronic way, upload it to the database, and the same day you collect the data, be able to actually look, compare it to with your previous year data or other material in your program, and make decision if you need. So in 2010, when I first walked the field before and in anticipation of what kind of fruit I'm gonna see in my hybrids, wishful thinking was the marker-assisted breeding, but now it became the reality since 2016. And so now for every single tree that I have in my field, I can almost attach like this label and say, okay, it has resistance to this, tolerance to this, potential to have this trait or that trait. So this is so exciting, and I'm so grateful to the community and the collaboration of all of the people involved in the research on rosaceous crops and prunus and peach per se. So right now, remember what I said with the brown rot, what I started with? This is in 2014, we found this individual through the genomic prediction to be brown rot tolerant, and this one to be bacterial spot resistant. And in the meantime, with the new marker, we determined that this particular one is also tolerant to bacterial spot. So yay, success. Not easy, but you can get there. Of course, none of this would be possible with the numerous funding from the federal agencies, from support of Clemson University Experimental Station, South Carolina Peach Council, and Department of Agriculture. And without support of so many people, I could not list them because the 10 slides would not be enough. There are some pictures that just show some people that went through my program and through my lab. This is my current crew now, and it's growing because we just had a new postdoc joining us from Spain. And breeder's job is easy when you have support of so many people. And what I present here is just in the background of me stand so many people that went through the program and helped. And the start of the program, as I already mentioned, I have to thank Dr. William Oakey for his support and help, Dr. Zrichi and Werner from North Carolina State University that were so happy that Clemson is starting breeding program so they can actually have their selection have life after they close the breeding program for peaches in North Carolina. And Dr. John Clark from University of Arkansas and now Margaret Worthington because we are collaborating tightly, evaluating materials in each programs and sharing the pollen and knowledge and ideas. So this was what I had. I probably was a bit too long. Thank you very much for your attention. And if you have any questions, I'm open and willing to answer.
Thank you so much, Dr. Gasek. It was an amazing presentation. We really enjoyed it. And we do have some questions. The first one is, in terms of breeding for fresh fruit and industry, are you focusing more on one or the other? And how about nectarines? And do they have same challenges that peach has when breeding for elite cultivars?
That's an excellent question. So you have to put it into perspective of being a breeder for the industry that knows what they want. You know, so what they want was a peach, yellow flesh, freestone, melting. But in my breeding and in my material, I find so many great non-melting cultivars. So I always keep them. And whenever we have a showing, I always kind of put them there and show them, look, this is a great one and it has all these great qualities. So my focus is not narrowed down. I have all of the options on the table, so to speak. I also have several nectarines in the program that I really like. One of the problem with nectarines is that our environment is so unforgiving because of the fuzz on the skin protects the peach from so many damages and blemishes. And nectarines don't have that protection. So many great nectarine cultivars released in other programs, when we grow them in Southeast, they become blemished and blotchy, and nobody, I mean, the quality is there, but visually you would not ever pick that fruit to eat. So I have few selections that look excellent, and few of those from Margaret's program that I was growing the hybrids in my program and evaluating. We actually have a few really good ones from Arkansas program that I need to talk to Margaret, of course, and then work on releasing, on deciding what to do.
Thank you. We have another question, and it asks, even though the growers requested resistant cultivars for bacterial spot targeting fruit, does the bacterial spot in the leaves have impact in overall fruit quality?
It does if tree defoliates. And in the cultivars such as O. Henry, which is a great cultivar, one of the ones that in post-harvest does not lose any of the properties and fruit quality stays excellent. But on the bad side, if the bacterial spot season is favorable for the development of the disease, tree completely defoliates in June or July. And then the tree, instead of amassing the nutrients and storing them for the next year flowering or development of flower buds, has to use those resources to push the new sets of leaves. That affects the quality and the productivity in the next year. So that's a big problem. One of the things with the bacterial spot is that there are cultivars that have either tolerance to leaf or to fruit, but there are those that have tolerance to both or susceptibility to both. So those two are not linked, and we are trying to find the ones that will tolerate the leaf, but be completely resistant and blemish-free for fruit.
Oh, thank you. Here's another question. As parents, have you used recombinant inbred lines or open pollinated varieties?
Okay, excellent question, and yes for both. But in which cases for open pollination, you know, in 2017, when we lost the production in South Carolina, as I showed you where the magic happens in Clemson, because we were surrounded with lakes, we were not damaged as much. So of course, anything I emasculated was gone, because when you emasculating peach is the perfect flower. So you remove all of the parts of the flower, you expose the ovary. Any temperature below 32 degrees, that ovary is gone. So any of the crosses I made were gone. So I said, okay, I'm not gonna, lose the year. I'm just gonna take the open pollinate, anything that's left, basically, collect from the mother trees that I like, and then let's hope something comes up later on in evaluation. As for the recombinant inbreed lines, there is an excellent breeding program in Spain, and they're basically trying to do the introgression from almond, because there are so many traits in almond that we can really use in peach. And they develop recombinant inbred lines. They sequence the genomes of all of these lines, and they know exactly which portion of the almond genome is inserted in the peach and vice versa. And so I'm very good friend and collaborate with them. And so we are discussing about sharing the pollen, and me being able to use it in my program.
Thank you. Another attendee is actually interested in, she has asked a question, if breeding in that nice lake surrounded area could be a problem, will the selected varieties grow somewhere else where the environment is not very suitable?
Oh yes, that's exactly why we are doing the replicated trials. So I mean, don't get me wrong, that nice area is nice, but we also get frost, and we also need to have a wind machines, and I built a tent first year. So one of the stories, we built a tent, we bought the heaters, we put the heaters to heat the tree. Next morning we come and wind blew the tent and everything froze. And so, so the trees that were not under the tent actually failed better than the one that we covered with the tent. So there are all kinds of stories that I believe every breeder can tell you. But that's why we need to do the replicated trials in different environments, because you never know what detrimental trait can come up on the surface in different environments. And if you imagine for the growers that have to plant the orchards, they put amount of money into establishing the orchard. They wait for 3 years till they get the first fruit. They cannot just sit and wait and not do anything. They have to apply all the things in the orchard to spend the money. And then if in the 3rd or 4th year, when the fruit is finally there, something horrible happens, they lost all of that money. So that's the reason why it lasts 15 to 20 years.
Thank you. And there's one more question. And Joshua is asking, hey Xenia, has your transition to an outdoor job as a breeder been what you expected?
Oh yeah, I love it. I mean, I forgot to mention that I am actually unique. I am 100% research and 12-month employed, and I have an office and lab at campus, and I have office and lab at the farm. So I go when the tree is cold. I don't have to sit and wait because I have to teach or anything. I have my students, we do the research and I mentor them, and I do one-on-one kind of a teaching. And I had 2 years endeavor in playing into teaching a plant breeding course. But this way, when the season is right, when the things are right in the field, I'm right in the field, and I just love it. During the summer, that's where I am. I'm outside. Heat, humidity, no problem, because peaches are there. And so this thing behind me is the first cultivar that's going to be released from the program. It's the first one that we planted in the replicated trials, and growers love it in Alabama, love it in South Carolina. And so Margaret, I have to thank and give kudos to Margaret for helping me understand the patenting process, because that's a huge new area for me. And for the Clemson, because there was no breeding program before, so we did not know what to expect and how to work with that. And so Margaret is on my speed dial, so to speak, with all of the questions.
Thank you. And we have one last question. So one of our attendees is curious of how many people or how much time does it take for you to perform all the crosses and evaluate the peaches each year?
Okay, that's a great question. As many as I can get. So bloom time in South Carolina, if the year is what I would wish for, is about March, mid-March. That's the period of time when students are on a spring break. So it's hard to get undergraduates to stay. They just go around the country and enjoy their free week. But any colleagues and any surrounding labs, we just say, okay, anybody wants to go outside, enjoy the beautiful weather and do emasculation, and we just get anybody. For pollination, we keep it to our trained crew, because if you can imagine what I do, I emasculate the whole tree, and then the stigmas are receptive for pollen within 72 hours. So within the window of 72 hours, I can decide when I pollinate.
Wow.
And we always try to wait at the end, because if you want to say it in a layman terms, the stigmas become desperate, to get where's the pollen, because, there's nothing coming. And then when we apply even maybe the pollen that they would not accept, which is self-compatible, and so that's not that big of a deal, then usually the efficiency of pollination is really high. So when we start pollinate, when you emasculate, it's visual. You see open bud or flower, it's easy what's emasculated, what not. But when you pollinate, you don't, pollen is tiny grains. So what we do, we just shake the pollen grains, deposit on our finger, and go and tap each. So there's about 3,000 flowers on the tree that you have to go, and tap and pollinate. It takes about hour or two with 5 to 6 people to do the big tree, big open center tree. So it's a kind of, ladders and climbing and all of that. Pollination goes quickly, because that, even though it's tedious and you have to pay attention what you pollinated and what you didn't, it goes much faster. And then what I also don't do, unless the cross is for the genomic studies, I don't cover or protect the tree after I pollinated it. So there is approximately 5% chance that the pollination will happen with the pollen that's not what I wanted. But, as great Dr. John Clark said, breeding is art, and there's a lots of serendipity going on. So if something happens without me knowing, I don't really care if the cultivar or the resulting is the cultivar or the combination of traits that I want.
Thank you so much, Dr. Gassick. It was an amazing moment to get to hear you.
Thank you.
Thank you, Dr. Gassick. Okay, that concludes our presentations for this Plant Science Symposium. I wanted to thank all of the speakers for their incredible presentations, and I would also like to thank our attendees for logging on from around the world to participate in this symposium. And that does mean so much to us with the Plant Science Council here at the University of Florida. So as promised, I'm now going to go ahead and reveal the winners of Thursday's breeding game competition. But before I do, I would like to thank Dr. Rex Bernardo for developing the software and taking time out of his day to moderate the session. So normally we would be on stage right now and everyone would clap as I announce your names, but obviously that's not going to be happening today. So I'm just going to go ahead and read off the winners, and then perhaps at the end, we can all give a round of applause, or just go ahead and do the clapping emoji on Zoom. So with that, I'll go ahead by announcing our third place winners. They are Gabriel Matsumoto, Joshua Anderson, and Gerline Kaur. Our second place winners are Beatrice Gouveia, Rafael Yasu, and Ryan Cullen. And finally, our first place winners, who developed the highest quality craft beer wheat, if you will, are Ronald Tapia, Lucian Gamir, and Leonard Herndon. So if you all can join me in giving them a nice round of applause, that would be fantastic. Thank you, thank you, Jason. So to our winners, I will be in touch via email to determine the best way to distribute your prize money. And I'm actually now going to go ahead and pass it off to Adina Grossman, our poster chair for the Plant Science Council. And she's going to go ahead and announce the winners of this year's poster competition.
All right, thank you, Julian. Well, this year we had a great variety of plant science research that was presented during the symposium poster competition. I'd like to thank all the judges for taking the time to score each poster, and to everyone who participated in the Q&A session yesterday and asked the presenters such engaging questions about their research. And of course, thank you to the poster presenters for doing such a great job with their posters and adapting to the online format. Now, I'd like to announce the 3 winners of the poster competition. If I call your name, please stay on the Zoom link after Julian concludes the session so we can take a group picture with the student winners. In 3rd place, we have David May. In 2nd place, Jordan McBreen. And in 1st place, we have Andrew Egesa. Congratulations to the winners and to all the presenters for doing such a great job. And please remember to stay on the Zoom link after Julian concludes the session.
Okay, thank you, Adina. So I would like to begin these closing remarks by once again thanking all of our invited speakers for agreeing to share your knowledge and experience with us. I want to thank all of us, everyone who registered for this event and participated in this year's symposium. Lastly, I would like to thank Corteva for allowing us the opportunity to put this event together, because it's events like these that forge the connections and grow the knowledge that we as plant breeders and plant scientists will be using for the rest of our lives. I don't think I need to reiterate how tough this year has been for many of us. Some of us weren't allowed in our labs for months. Others, which I learned of yesterday at our networking session, were only back allowed in their labs a month ago. At the start of this pandemic, I remember thinking, I will still be able to plan the symposium for in-person. You know, it's so far away, I won't have to worry about that. Yet here we are nearly a year later with a virtual symposium. There were some positives to this online format. We were able to invite speakers from across the country to present their research to us. We were able to invite attendees from around the world to listen in on these talks. Science communication is changing, and some of these changes may remain long after this virus is gone. Over the last 2 days, we've learned about starting a breeding program from scratch and how to deal with the pressures of taking over an established, successful one. We've heard about growing plants for our eventual transition to the stars and growing crops for malnourished and underrepresented countries here on Earth. We've listened to talks about what it means to work in a hugely successful breeding company such as Corteva, and what it means to run your own hugely successful breeding company here at UF. Lastly, we've learned from our fellow graduate students as they take on this journey of a PhD and they share their research and experience with us as we sit back in awe at the progress they've made. Maybe that last part's just me. I've actually thoroughly enjoyed sitting at my desk here for the last 2 days and being a part of this symposium. And now that it's over, I kind of have a sense of sadness. I'm not really worried about it though, because rain or shine, there will be a 6th annual Plant Science Symposium next year. And I hope to see you guys all there next year as well. So with that, again, thank you all for joining, and have a great rest of your afternoons. Thank you. And again, as Adina mentioned, if you did win the poster competition, please stay on a little later so we can go take some pictures. Okay, Adina, do you want to take over for the posters?
Sure. We just have to get the 3 winners as panelists so they can share their turn.
David just replied that he's not being able to access his camera or video. So
Is it because he's not a panelist?
Yeah.
Oh, there he is.
Oh,
Okay, so we have to make.
I think the best solution, and I don't necessarily need you, right?
sorry, what, Gabe?
Like, I think you can become attendees, so only the poster winners would be here. I don't know.
You can just turn your camera off, Gabe.
Is Andrew here? I'm looking in the attendees and don't see him. Oh, he's a panelist. Awesome.
There we go.
Okay, so everyone's here. I'll turn off my camera. Modesto, do you want to take a picture?
Sure, I'll do that.
Okay, and congrats, guys.
Oh, Jason, we can't hear you. Still can't hear you. You can type it in the chat if you like, and I could read it out. Thanks, Jason.
Thank you, Jason.
Okay, congrats, guys. We will be taking the pictures in 1, 2, go. One more. Oops, give me a minute.
Guys, I have a quick note to say. I mean, maybe it would look kind of weird with us turning off the video, and then some photos and then some videos would look patchy. Either like we could leave and then only Modesta would be there to take the photos, or either all of.
I don't see you guys. Don't worry. It's just the winners that I see.
Oh, okay, cool.
Fine. So 1, 2, 3, go. Perfect. Congratulations, guys. Yeah,
Yep,
thank
congrats, guys.
Okay, thank you, guys. We're going to reach out to your emails about the prizes. So just expect an email from me and the other members.
Congratulations again.
Good job, guys.
Thank you guys so much. Thank you.
Thank you for participating.
Thanks, you guys.
Okay, I'm going to go ahead and end this session here. So.
All right, congrats, Julian and everyone.
Yeah, thank you guys. Thanks, Julian, Adina, Gabe, Modesta. You guys did amazing.